So here we are in chapter 13. Hopefully it'll be a little more interesting than chapter 12 because chapter 13 is all about our senses, and we'll probably spend a lot of time on the eye and the ear, and that's what we're going to focus on. This is the part A PowerPoint. This gives you kind of how they've divided chapter 13 into these four parts. We're going to go over the eye, the ear are our main ones, and then we'll talk about reflexes at the end. So if we can get through part of the eye tonight, that's my goal. We'll kind of review the anatomy for it, and then we'll tie it all together and learn how we see and things like that. So here we have the place of the peripheral nervous system and its structure. Remember, in the peripheral nervous system, we have a sensory division and a motor division. In the sensory division are all of our senses, and that's what we're focusing on in this chapter. A lot of our senses have to do with sensory receptors. So the receptors, the ascending pathways that bring up those stimuli from those receptors, and then how the cerebral cortex processes all that sensory information. So remember, all of your sensations and stimuli, whether it's coming from nerve endings in your skin, the photoreceptors in your eyes, hearing receptors in your ears, they will all get sent usually to the thalamus in the brain. And then the thalamus will send out which part of the brain is responsible for decoding what sort of sensory information is coming to it. So our survival depends on sensation, which is the awareness of any sort of stimulus, whether that's an internal or an external change. And then perception, which is just what the brain does, an interpretation of the meaning of that stimulus. The somatosensory system is the part of our sensory system that serves all the body walls and limbs, and it receives inputs from exteroreceptors, proprioceptors, and interoreceptors. We'll talk about what those are. Input will be relayed towards the head, but it can be processed along the way, whether that's in the medulla oblongata, right in the spinal cord, or in the thalamus region. Levels of neural integration, we have the receptor level, where we'll have your sensory receptors, the circuit level, where processing will occur in any ascending pathway, and all of those pathways are kind of tracks or columns within the spinal cord. That's what the second half of the chapter 12 lecture that I posted on Tuesday. And the perceptual level is the processing in the cortical sensory areas, which is in the brain. So this takes you through processing the sensations at the receptor level and how they will generate a signal. So for a sensation to occur, a stimulus must excite a receptor, and an action potential must reach the central nervous system, whether that's the brain or the spinal cord. Transduction must occur, and that just means that the energy of the stimulus will be converted into some sort of action potential eventually. And an action potential means the nerve has fired and will take that stimulus information to the brain. Adaptation is change in sensitivity in the presence of constant stimulus. So if your senses or one sense receptor is constantly being bombarded with the same type of stimulus, those sense receptors will eventually adapt, where the receptor membrane will become less responsive, and eventually the action potentials will decline in frequency or stop. And I should have put this in here, but a great example of this is in lab, I'll show you a green circle around a red dot. And I think I did this in anatomy. And if you stare at the red dot in the middle, eventually the green circle around it disappears because your eyes have adapted. And the sense receptors in your eyes have kind of stopped perceiving that stimulus. We have phasic receptors, which are fast adapting, and tonic receptors adapt slowly or not at all. Nociceptors are pain receptors, and proprioceptors give perception of your body and generalization with other body parts and tendons. This talks about how your senses are processed at the circuit level, and it talks about the pathway of 3 order neurons, which take that sensory information to the brain eventually. So this is just processing at the circuit level. And if I skip over this or go over it relatively quickly, I won't ask you details about it. And then processing at the perceptual level is interpretation of the sensory input. Will depend on the specific location of target neurons in the sensory cortex. So the exact point in the cortex that's activated will be always linked to activation of the same sensory neuron. So what this means is that sensory information will go to your thalamus, and your thalamus will project that sensory information to a specific place located on the brain. I'm going to skip over that for now. Perception of pain. So we'll talk a little bit about pain, and again, eventually I'm going to try to get to eye if I can. So I might go over these first couple slides quickly. Pain is warning of actual or impending tissue damage, so protective action can be taken. So pain will be important to tell your body to start kind of bringing in the soldiers or the army to help if something is about to be damaged. Some pain impulses are blocked by inhibitory endogenous opioids like endorphins. Pain tolerance, we all perceive pain at the same stimulus intensity, but the pain tolerance will vary. Sensitive to pain means low pain tolerance, not low pain threshold. And your genes, this is actually genetically determined, will help determine your pain tolerance, as well as your response to different pain medications. Some pain medications work well on some people, some do not. Visceral and referred pain, this is important to know. I talked about this when I taught anatomy too. Visceral pain results from stimulation of visceral organ receptors. So you might feel a vague aching, gnawing, or a burning. So something with your viscera or internal organs is wrong. Referred pain is where pain from one body region is perceived as coming from a different region. And this is because the visceral nerve fibers, which are coming from the organ that is damaged, such as your heart, and your somatic pain fibers that are coming from the skin receptors in a part of the body, they will kind of travel along the same pathway up to the brain. So the brain assumes that the stimulus is coming from the common somatic region on the skin. And the best example of this is how if someone's complaining of extreme left arm pain, that's referred pain for a heart attack or some sort of myocardial infarction where there's damage to the heart tissue. And this is a map of referred pain. You can see here how pain in the heart will be referred to the entire left side of the left arm. For some reason, the lungs, diaphragm, gallbladder can be referred up to the right shoulder. And again, this is because somatic sensory receptors, which are coming from the areas on the skin that are highlighted here, they'll travel along the same pathway as the visceral sensory receptors coming from your internal organs. So again, extremely important in the medical field, referred pain. There's also phantom limb pain, which is pain felt in a limb that has been amputated. And some anesthesia is used during surgery to reduce that phantom pain. So common during amputation. All right, so sensory receptors are specialized to respond to changes in the environment, and we call that a stimuli. Their activation results in a graded potential, which is kind of a localization of depolarization that will then trigger a nerve impulse. Sensation is awareness of a stimulus, and perception is the interpretation of the meaning of that stimulus. And we can classify receptors by their type, where they're located in the body, and their structural complexity. So mechanoreceptors are often found in your skin. These receptors respond to touch, pressure, vibration, stretch. Thermoreceptors, also found in the skin, are sensitive to changes in temperature, also are found internally as well. Photoreceptors respond to light energy, so these will be located in the retina, the back of the eye, and chemoreceptors will respond to chemicals like your smell, taste, changes in your blood chemistry. Nociceptors are sensitive to pain, extreme heat or cold, excessive pressure. Nociceptors will often stimulate other thermoreceptors and mechanoreceptors and chemoreceptors. Then they're classified by location. So you can have an exteroreceptor, which will respond to stimuli from external, outside the body. So these will be touch, pressure, pain, and temperature. And most of your special sense organs will be exteroreceptors, the ear, the eye. Interoreceptors, also known as visceral receptors because they will respond to the viscera or the organs. Also blood vessels, so they'll be sensitive to chemical changes, tissue stretch in the blood vessels. Especially important if you're dealing with someone with high blood pressure, their tissues will obviously stretch more. Temperature changes, sometimes they can cause discomfort, but usually the person is unaware of the interoreceptors working. And then proprioceptors will respond to stretch in your muscles, your tendons, your joints, your ligaments. They will inform the brain of one's movement. So proprioception is just the idea that your body parts know where they're all located in relation to the other. So your legs aren't beating up against each other when you're trying to run, or your arms aren't hitting each other when you're trying to walk. So that's what proprioceptors do. They inform the brain of one's movements. Classification by receptor structure. Simple receptors of the general senses. These are just dendritic endings. They're found throughout the body. Then we have receptors for special senses, and these will be in your eye, your ear, hearing, equilibrium, smell, and taste. And all are housed in complex sense organs. We'll get to that in chapter 15, but we're going to get over some of the physiology of those. General senses include your touch, pressure, stretch, vibration. A lot of these are found in your skin. Receptors have either non-encapsulated nerve endings, which are free, or encapsulated nerve endings. And the difference between them are free nerve endings will just be like ends of nerves that have branched off and have ended. And encapsulated nerve endings will be in their own separate structure. And we'll talk, the free nerve endings are abundant in all of your epithelial and connective tissues. They could be thermoreceptors located in your skin, nociceptors, which will detect pain a lot in your skin. These can also be itch receptors in your skin. Then we also, free nerve endings could be tactile Merkel discs, hair follicle receptors. For an example of that is allowing you to feel a mosquito landing on your skin. So these are examples of non-encapsulated nerve endings. Encapsulated dendritic endings. These are mechanoreceptors whose terminal endings are encased in a connective tissue capsule. So they're not just free nerve endings, they're encapsulated. We have Meissner's corpuscles, important for discriminative touch, so they'll be in your fingertips. Pacinian corpuscles are responding to deep pressure and vibration, and they're located deeper in the dermis. Ruffini endings, muscle spindles, tendon organs, and other joint receptors are just other examples of receptors located throughout the body. And these are some examples there in their functional class and body location. Okay, so then we'll get to the eye a little bit. Some 70% of body sensory receptors are in the eye, and that's because your eye contains so many photoreceptors, which are your rods and your cones. It's a small sphere, only 1/6 of the surface is visible. That's what you see in the sclera and the pupil and the iris. Most of the eye is enclosed and protected by a fat cushion and a bony orbit. So you have several bones, your zygomatic bone, sphenoid bone, that all create an orbit for your eyeball to sit in. And the eye itself will consist of the accessory structures and the eyeball, which are all shown here. Hopefully some of this is a review from anatomy, the accessory structures and the eyeball itself. Here are accessory structures, your 6 eye muscles. The lacrimal apparatus produces tears. The conjunctiva is a thin membrane on the inside of the eyelid, the eyelids and the eyebrows. So here are your eyebrows. They shade from sunlight to prevent perspiration from reaching the eye. The eyelids are also called palpebrae. There's a slit or fissure where your eyelids will join when you blink. The lacrimal caruncle is at the medial commissure, contains oil and sweat glands. So I'm going to skip over the eyelids. They'll blink reflexively every 3 to 7 seconds. Try to have a staring contest with someone and know that every 3 to 7 seconds they'll have to blink. Eyelashes also help to kind of keep things out of the eye. Tarsal glands help to lubricate the eye, and you have ciliary glands that are modified sweat glands. So here's a look again, eye and accessory structures. A STI is a painful inflammation of any of the sebaceous glands. A chalazion is an obstructed tarsal gland that may result in a firm or painless bump. The conjunctiva is the transparent mucous membrane. It produces a lubricating mucus secretion, and it'll all fall kind of on the inside of the eyelid. And I won't ask you guys to know any other things than that. Conjunctivus, it's an itis word, it's inflammation of the conjunctiva, and pink eye is an infection of the conjunctiva caused by bacteria or viruses. Highly contagious. So with kids, daycare, if one kids get pink eye, usually they all get it. The lacrimal apparatus produces the tears from a lacrimal gland. The duct then will drain those tears into the nasal cavity. Lacrimal secretion is important because it has mucus, antibodies, and enzymes that help to keep your eye clean at all times. Blinking spreads those tears, and then the tears will eventually be drained through the lacrimal sac, the nasolacrimal duct, and into the nasal cavity. So that's why if you're crying, if you cry more, your lacrimal gland is producing more tears, and you will empty those tears into your nose, so you might need a tissue. That's why someone offers you a box of Kleenex if you're crying. Okay. I'm just going to skip over this clinical one. 6 extrinsic eye muscles. I'm going to skip over this. Hopefully you remember these 6 eye muscles from anatomy. They attach to the eyeball and they will move the eye in many different directions. Okay. All right. Cross-eyed is congenital weakness of the external eye muscles. So the eyes will be rotated medially or laterally, and you'll notice that in some people, and it's usually a problem with one of their eye muscles. They're losing, or they're weaker than others. Okay, so the wall of the eye, the fibrous layer, vascular layer, and inner layer, and an internal cavity with fluids called humors. The lens will separate the internal cavity into an anterior and a posterior segment, and that is seen here. So you can review this anatomy a little bit on your own, but we're going to focus a lot on the retina where your photoreceptors will be located. A review from anatomy, I'll take you through a little bit of this. Light will come in through the cornea, which is this transparent concave shape, and light travels through the pupil, which is the black part of the eye. It travels through the lens, and the lens will refract or bend the light to a certain point on the back of the eye, which is the retina. You can see here the anterior segment contains the aqueous humor, and the posterior segment contains vitreous humor. The anterior segment will be more a watery secretion, and the posterior segment, the vitreous humor, is a lot more like jelly-like. That gel-like structure in the posterior segment helps to give your eyeball the shape so it doesn't collapse on itself. So the white part of the eye is the sclera. The pink part, the second layer in, is the choroid. That's where your blood vessels will be. The choroid layer includes the ciliary body, which has suspensory ligaments, which are these little tiny thread-like structures that will control the shape of the lens. And depending on if the lens flattens or bulges out and becomes round, that will refract or bend the light that comes through to a certain spot on the back of the third layer, which is the retina. Within the retina, we have a macula lutea, which is an area with a fovea centralis in it. And the fovea centralis is where you have the highest proportion of cones. The optic nerve attaches to the back of the eye at the optic disc. And the optic disc is called the blind spot because there are no photoreceptors here. You can see the optic nerve carries all the blood vessels into the eye. So there's no area for photoreceptors at the optic disc. So this takes you through kind of what I just talked about in a little more detail if you want to review the eye on your own a little bit. The iris is the colored part of your eye, and it contains 2 smooth muscles, types of smooth muscles that will control how the pupil constricts or dilates. Changes in emotional state, the pupils will dilate when the subject matter is appealing or requires problem-solving skills. So if you're in a fight or flight sympathetic response, your pupils will dilate to try to take in as much light as possible. So again, another look at the eye. I think that's pretty similar to the one we just saw. Here's a parasympathetic response. Specifically in the iris, the sphincter pupillae muscle will contract, the pupil will constrict and decrease in size. Remember, your parasympathetic is your rest and digest division. So when you're just resting, digesting, your eyes don't need to take in a lot of light because they're not thinking or perceiving anything. The sympathetic division, the dilator pupil muscle contracts, causing the pupil to dilate. So the size of the pupil increases. And again, your sympathetic is your fight or flight response. So if you're in a really stressful situation, your pupil will dilate to try to take in as much light as possible because you're trying to perceive as much around you as you can. And you can kind of see here how these muscles are arranged. Again, the iris has two muscles, the sphincter pupillae, responsible for the parasympathetic, and the dilator pupillae, responsible for the sympathetic. So that's your iris. We all have different colors, but they all work the same. Then we get to the retina, and this is what I want to talk about because the retina is where the magic of seeing happens. The retina contains millions of photoreceptor cells that transduce light energy. And remember, this word transduce takes the stimulus of light or touch or smell, but transduce takes light in the eyes and transduces it to electrical energy or changes it to electrical energy. There will be neurons, glial cells. There's an outer pigmented layer and an inner neural layer. So here's our retina. You can see here the neural layer. The pigmented layer is the outermost. So the pigmented layer will be where all of your photoreceptor, ganglion cells, horizontal cells are located. And the neural layer will be where all of the axons from this optic nerve branches are. So we have the pigmented layer, which will be all of your pigments. So your pigment proteins in your cones, and your neural layer will be all the axons of the optic nerve. Real quick too, the pathway of light comes in this way and will bounce off the pigmented layer first and kind of work its way back forward to the neural layer. So the pigmented layer, the functions of that will be to absorb light, phagocytize photoreceptor cell fragments after they've been used and storing vitamin A. The neural layer then is the transparent layer. I'm sorry, I misspoke here. The neural layer is what's composed of your photoreceptors, your bipolar cells, and your ganglion cells. The signal will spread kind of from back to front, from your photoreceptors to your bipolar cells to your ganglion cells. And then the ganglion cells will their axons will exit out of the eye as your optic nerve. The optic disc is where the optic nerve leaves the eye, and there are no photoreceptors in this spot, so we call this your blind spot. And we call it your blind spot because as your eye is constantly moving, it's trying to focus things on the retina to avoid this blind spot. But there are different tests you can do, kind of peripheral vision tests where you can see as you're looking at an image, if you focus on it, there will be a part kind of about 5 inches from your face where that image will lose focus and it'll disappear, and that's referred to the blind spot. The retina has 250 million photoreceptors that are one of two types, rods or cones. And you can see the rods and the cones will be located here in this neural layer. The pigmented layer, as you can see, is this kind of pinkish, really thin layer between the choroid and the neural layer. The rods, the numbers are greatest at the periphery. There's no color vision or sharp images in rods. Your rods are about 20 times more numerous than your cones. So you have more rods in your retina. They help with dim light and peripheral vision. Your rods are really important. If you go into a room that's really dark, it'll take about 15 minutes for your rods to adjust and try to absorb as much light as possible. And that's why after about 10-15 minutes, your eyes can adjust and see better in a dark room than the initial entrance to that room, because your rods are just working on taking in as much light as possible. Your cones are the vision receptors for bright light and color. The macula lutea is an area at the back of the eye. It's lateral to the blind spot, meaning lateral to the optic disc. It will contain mostly cones in it. Specifically, these cones will be located in the fovea centralis, which is about the size of the end of a pin or a needle. It's a tiny pit in the center of that macula. It will contain all cones, so is the region with your best visual acuity. And again, your eye movements will allow you to focus on an object so that the image is usually directed to land on the fovea centralis, which is just the area of highest cones. So here's a look at the neural area. So again, here where my cursor is the posterior cavity. So when light comes in, light actually goes all the way to the back of the eye first, to this pigmented layer, and then light will be picked up by the photoreceptors, whether they are the rods, which are in bluish, or the cones in yellow. And again, the rods are more numerous than cones. Then light will be transduced into electrical energy as it passes through bipolar cells, ganglion cells, and eventually to the axons. And these axons of these ganglion cells will take those light stimuli back up through the optic nerve. So again, light comes in, it goes all the way to the back of the retina first, and then it works its way back superficially to these axons of the ganglion cells. This is a photomicrograph of the retina, so it's kind of fun to see this in real life because you're like, oh, this actually does exist. Some guy just didn't make it up. So here's showing the choroid layer, the pigmented layer, the outer segments of rods and cones. Here's the nuclei of those rods and cones, bipolar cells, and then the ganglia are shown here. And this is the axon of those ganglion cells. So kind of cool to see it in real time. Let's see. Two sources of blood supply to the retina. These are your choroid vessels that are supplying the photoreceptors. And all of these blood vessels, again, that are providing blood supply to the retina, just enter and exit the eye in the center of the optic nerve. These vessels are visible in a living person. So this is a picture of your retina that your eye doctor might take a look at. So here's the macula lutea, which is directly lateral to the optic disc. So I would assume this is a picture of the right eye. Here's the optic disc where the blood vessels are coming out of. So you can see the central artery and vein. And again, the optic disc is the eye's blind spot because there are no photoreceptors located at the optic disc. A lot of cones will be located in the macula lutea. Retinal detachment, you might hear of this. It's a condition where the pigmented and neural layers will separate, allowing that jelly-like vitreous humor to seep between them. And this can lead to permanent blindness because if your retina detaches from the pigmented layer, your retina has no way of getting any blood supply to it, oxygen. Usually happens when the retina is torn during a traumatic blow to the head or sudden stopping of the head during movement. So maybe don't go bungee jumping, boxing, I don't know, tackle football. I'm not a big fan of any sort of contact sport because I think it just does a lot of stuff to your body that maybe you don't need to go through. But no judgments though. What else? The symptoms for that are described as victims as a curtain being drawn across the eye because they're basically just losing complete eyesight. You can reattach the retina with laser surgery. Internal chambers and fluids, more anatomy here. The posterior segment and the anterior segment are separated by the lens. Again, going through what makes up the posterior segment, that vitreous humor. It will transmit light. The anterior segment contains more of the aqueous, watery type humor. That's all I want you to know about those kind of segments. Again, the lens separates out the anterior kind of chamber from the posterior chamber or segment. Okay, Glaucoma is a condition in which drainage of the aqueous humor is blocked, causing fluid to back up and increase pressure within the eye. So really simply put, glaucoma is just pressure buildup within the eye due to some sort of drainage system not working properly of that aqueous humor. It can increase to dangerous levels, which could compress the retina, the optic nerve, leading to blindness. There are a few early signs to this, which is unfortunate, but late signs include seeing kind of like halos around the lights and blurred vision. So you can get tested for it. Treatment are some eye drops or laser therapy or surgery. The lens is a biconvex, transparent, flexible, avascular. Let's just go to a picture. The lens can cloud up due to consequences of aging, diabetes, heavy smoking, or frequent exposure to intense sunlight. Some are congenital. Vitamin C increases cataract formation, which is kind of interesting, but the lens you can replace your lens surgically with an artificial lens. And I'm just gonna go ahead and exit out of this and we'll start part B. We'll get through that as much as we can. Any questions so far? Just on a scale of 1 to 10, put in the chat how confused you are, if you're still with me or awake. 10 being okay with it, 1 being kind of confused. All right, and what we're gonna do is we're gonna just try to get through the eye tonight. And then we'll kind of get through the ear and taste on next week too. Okay, so let's talk a little bit about eye and seeing. I'm gonna go through, kind of skip over the wavelength and colors that your eye sees, and we'll kind of start with refraction, because refraction is how your eye will bend the light and focus it on the back of the retina. So refraction just means bending of light rays. And the lenses of the eyes can also refract light because they're curved on both sides. A convex lens means it's thicker in the center, and concave means it's thicker at the edges because the lens will change its shape a little bit. Also, the liquid within your eyes can also refract or bend the light rays. Convex lenses will bend the light passing through it so that the rays converge at a focal point, and the image formed at the focal point is always upside down and reversed from left to right. So this is interesting. All the images that are coming into your eye, the lens will turn them upside down and kind of switch them so they're backwards. It's your brain is what turns it right side up so that you are perceiving something right side up as you're looking at something. So here's just a look at how the lens, so this is a image being inverted, upside down, and reversed. So this is an example. We're just going to focus on here. So this is like a 4-quadrant squared, and you can look at the colors. And this is how your lens will invert it and also change it from right to left. And that's how the lens just kind of flips things around to be perceived on the back of your eye. Light is refracted 3 times along the path as it enters the cornea, enters the lens, and leaves the lens. And this is the pathway of light entering the eye. Kind of a review from anatomy, but I want you to know this. It goes through the cornea, the aqueous humor, the lens, the vitreous humor, the entire neural layer of the retina, and finally your photoreceptors. Focusing for distant vision. Eyes are best adapted for distant vision, and the far point of vision is the distance beyond which no change in the lens shape is needed for focusing. In order to focus on something distant, the ciliary muscles are completely relaxed in distant vision, which causes a pull on that ciliary kind of body, the zonule, as a result. So lenses will be stretched flat in order to help see things far away. So this is just a look at how the lens, the ciliary muscles, and the ciliary zonule. So this is just, it's the ciliary body that's connected to these suspensory ligaments that will control the shape of the lens. The ciliary muscle contraction will loosen the fibers and relaxation tightens them, just depending on whether it's trying to focus on something far away or near. So takes you through what will happen, how the lens will flatten for distant vision. Sympathetic input will relax the ciliary muscle, and it kind of is opposite in how you're thinking of this, but when that ciliary muscle is relaxed, that actually tightens the ciliary zonule, which are those suspensory ligaments, and that will flatten the lens. So the tightening will flatten the lens, and this helps you see things distant, so things that are further away. For close vision, we call this accommodation of the lenses, where the lens will change shape to increase how light gets bent as it comes into it. Um, Presbyopia is the loss of accommodation over age 50. So this is why many people over 50 need reading glasses. The near point of vision is the closest point on which the eye can focus. Focusing on close vision, constriction of pupils, your pupillary reflex will constrict the pupils. The convergence of the eyeballs, they'll medially rotate, causing convergence of the eyes towards an object being viewed. And in terms of the lens, when you're trying to focus on something close up, parasympathetic input will contract the ciliary muscle. This will loosen the ciliary zonule, or the suspensory fibers, and this will cause the lens to bulge. So I know this is a little confusing, but this slide right here, I want you guys to understand the difference, what the lens does for looking at something close up. It will bulge for close vision. And for looking at something further away, the lens will flatten for distant vision. And I just want you to know, and the best way are probably just these two slides, letter B and letter C, how the lens does that, to understand what shape the lens will take when you're looking at something close up or furthest away. Clinical problems, myopia is what most of us are who need glasses. We're nearsightedness, so which means you can only see things near and it's harder to see things farther away. Hyperopia is farsightedness, meaning it's harder to see things close. And astigmatism is when you have unequal curvature in different parts of the cornea or lens, and that's usually corrected with a contact lens or a laser procedure. Functional anatomy of the photoreceptors. So these are your rods and cones. There's an outer segment and an inner segment. They're your rods and cones. The outer segment receives the light. This will contain your photopigments that will change shape as they absorb light. And the inner segment of each will join the cell body. So this kind of just takes you through some of the anatomy. Here's the outer segment of the rods and cones and the inner segment. And eventually these rods and cones will synapse or connect with a bipolar cell. The photoreceptors, more anatomy, there's visual photopigments embedded in discs. And these pigments will just change shape as they absorb light. Photoreceptors are vulnerable to damage. They can degenerate if your retina detaches. They're destroyed by intense light. So you don't want to look at the sun because that is damaging the photoreceptors. Comparing a little more rods and cones, rods are very sensitive to just light, making them best suited for night vision. They contain a single pigment, so the vision is perceived in gray tones only. The pathways converge on rods, causing fuzzy, indistinct images. As many as 100 rods may converge into one ganglion. So just remember that many rods will converge on one ganglion versus cones. Cones have low sensitivity, so they require bright light for activation. They react more quickly than rods. They have one of three colored pigments, and they do not converge. So that means that some cones have their own ganglion cell. So that means that one cone converges on one ganglion cell itself. So you have a 1-to-1 ratio, basically, of cone going to a neuron. So that gives the cones the ability to be very precise in their vision and color, whereas rods are not precise in their vision. They're more suited for low light situations. So this is just a comparison of rods and cones. As you're studying rods and cones, I probably wouldn't ask you much more about them outside of this chart. So just knowing the difference, basic, of rods and cones. Color blindness is lacking one or more of the cone pigments. So the most common is red-green, which is either red cones or green cones are absent. So sometimes color blindness if you are colorblind, they could just have one cone pigment, whether that's red or green or blue, that's absent. So you might not see your reds very clearly. They'll look more like browns. So that's color blindness. These are visual pigments. They're proteins. Retinal is the key light absorbing molecule that combines with one of four proteins to form visual pigments. So these are just different visual pigments that are in the photoreceptors. I'm not going to ask you too many details. Phototransduction is just the process, so it's a transduction word. The pigment will capture a photon of light energy, which is converted into a greater receptor potential. So remember, transduction is always taking in a sensation and converting it to a action potential or some sort of electrical stimulus. Capturing light, rhodopsin is the pigment in rods. Phototransduction, the pigment is synthesized, it's bleached, regenerates. So this is just capturing light. And this shows you exactly how this pigment will capture light. And you don't need to know the details about this, but you should know that pigments within these rods and cones are what are capturing light. And rhodopsin is an example of that pigment. And I'm going to skip over some of this as well. Events of phototransduction. So again, how the light comes in. Basically what it's doing is it's changing shape of these photopigments, and that change in shape is what will open up channels, which will cause a localization of ions coming in, changing the light energy you perceive into some sort of electrical event. Information processing in the retina. This shows how light will enter in through the eye and travel down through these steps, 1 to 2. The light traveling into the eye will eventually be transduced into electrical event that will open up channels where ions will rush in. Remember from your action potential, when ions rush in, that causes a depolarization, which sends a signal down. So that's where we're talking about with phototransduction. Your rods and cones take in the light and then send on that signal down the bipolar cells and ganglion cells, eventually to the nerve axon, to the optic nerve. Details that are shown here, you don't have to know details, but just that general kind of pathway. Okay, I'm going to skip over this. Light and dark adaptation. Cones respond to bright light. Rhodopsin is what is sensitive. Light adaptation. This is talking about how visual acuity will improve over 5 to 10 minutes as the rod systems turns off, and the cones and neurons will rapidly adapt. Dark adaptation. Rhodopsin in your rods accumulates in the dark. So when you move into a dark room, rhodopsin will begin to accumulate, and that will cause sensitivity of you to perceive things in a dark room within, I said 10 minutes, maybe more like 20 to 30 minutes. And that's because rhodopsin, the photopigment in rods, is accumulating in the dark. Night blindness is a condition in which rod function is seriously hampered. And this occurs in elderly as you get older, so the ability to drive safely at night is impaired, and that's just due to rod degeneration. Okay, visual pathway to the brain. Let me just see how far I want to go here. Okay, let's just talk about the pathway to the brain, and I think then we'll be done, because then we'll start with smell on what is it, Tuesday? Today's Thursday, right? So let's talk a little bit about the visual pathway and processing to the brain. So we know our optic nerve comes out the back of the eye. Medial fibers from each eye will cross over at the optic chiasm. You remember that from the anatomy, where the optic nerves cross over, and they continue on as optic tracts, which means that each optic tract contains fibers from the lateral aspect of the eye on the same side and the medial aspect of the eye on the opposite side. So each carries information from the same half of the visual field. And I'll show you what I mean by that. Most fibers of your optic tracts will continue on to the thalamus. And from there, like everything, all sensory information goes to the thalamus. And then the thalamus projects that information to the primary visual cortex, which is in your occipital lobe, where conscious perception of visual images occurs. So this visual pathway, I'm just going to show this a little bit here. If you're fixated on a point for both eyes, parts from the right and the left eye will perceive different kind of perceptions around this fixation point. And I know it's kind of hard to detect here, but you'll notice that in your left, so if you're, this is a look at the brain from the bottom. So here's your right eye, and it's taking in the fields from this area, and your left eye takes in fields from this area. So basically what this is showing, that each eye, the right and the left, are each taking kind of both sides of the field of view into their eye. These are the optic tracts, and they will all cross over at the optic chiasm. And what that means is that eventually everything that's seen from just the right side of the body will eventually all go to the left side of the brain, and everything seen from the left side of the body will go to the right side of the brain. But what your eyes do is your eyes will take in both medial and lateral views on both the right and left sides of the body. I know that's kind of confusing, but just in general, both your eyes will take in medial and lateral views around a fixation point, but then at the crossing point, what goes on to the brain itself will be from only one side of that field of view. So this is a look kind of showing in anatomy where your optic nerves are, where the tracts will cross at the optic chiasms. And then the optic tract will continue that on to the thalamus. And you can kind of start to see your thalamus is located here where it says lateral geniculate nucleus. That's a specific part of the thalamus. And again, then your thalamus will project that sensation on to the primary visual cortex, which will be in the occipital lobe. Depth perception are where both eyes view the same image from slightly different angles. Your visual cortex will fuse these slightly different images, resulting in a 3D image, which leads to depth perception. And again, this requires input from both eyes. The loss of an eye or destruction of one optic nerve will eliminate true depth perception entirely. So this will also affect your peripheral vision on the damaged side. If neural destruction occurs beyond the optic chiasm after those optic nerves cross, then part or all of the opposite half of that visual field will be lost. And an example of what could damage this will be stroke, which could lead to blindness in the right half of the visual field. More visual processing. I think this visual processing will all occur in the occipital lobe, but that's all I'm going to talk about there. Is I just want you to know that the visual processing will occur in the occipital lobe. And I'm going to stop the PowerPoint here.